in the west and the south. So this is really our first go at this. So yeah, I think we did a good job. Certainly something we can build on in terms of the number of paddocks that we collected from. But in brief, I'm going to be talking today not only about the Queensland survey, but results across the northern region. So I'll jump a little bit between what we found in Queensland and what we found across the whole northern region. But the Queensland surveys, we did both winter and summer. So that's a bit of a point of a difference for us in the north. We do have both winter and summer surveys, whereas in the southern and western regions, they really have their winter surveys. In winter, we surveyed in 2016 and 17, and in total, we visited 197 paddocks. So when we visit these paddocks, not every paddock is going to have our target weeds present, but where they are present, we collect a representative sample that we then use in our screening. Then in summer, we surveyed in 2017 and 2018. We did 170 paddocks. The reason we've split these surveys over 2 years is purely because of the seasons that we encountered. For example, when we did the 2016 winter survey, we actually got some wet weather, which was nice, which meant that we couldn't survey the whole region because we got rained out. So we had to go back and revisit again in 2017. That's the reason why we've got surveying split over those 2 years. So just quickly going through our target weed species for the survey. You can see there we've got a number of species in the maroon and a number in the blue. Those that are in the maroon are the ones that we tested here in Toowoomba, and the species that are in the blue were the ones that were tested by John Broster down in Wagga. So you can see we've got all the key players there in terms of our key summer and winter weed species. Just quickly in terms of now the summer weed species, these are the ones that we tested here in Toowoomba, and you can see the range of herbicides that we did screen these weeds for resistance to. So again, we focus predominantly on glyphosate for each of our species, and then a few key herbicides that we thought that these weeds might be at risk of developing resistance to. So those herbicides that are commonly being used to control these weed species. I did want to point out that for Ornless barnyard grass, windmill and liverseed grass, we did screen for imazapic resistance. However, imazapic being a pre-emergent herbicide, we actually screened it as a post-emergent, so which is not in line with its use pattern unless it's in mixture with something like paraquat. So the results we got for imazapic, we need to, I guess, take with a grain of salt. So I'm actually not going to present in any detail the results that we got for that herbicide. I will mention them in passing. You can see though for the grasses in particular, we also focused on some of those group A grass selective herbicides as well. And then we introduced paraquat for feathertop rose grass as one of our other targets. Just wanted to give a bit more background or a bit more information on the approach that we used for the testing of the herbicides, just for those who may not be familiar. So when we've collected samples out in the field, we've then germinated those on agar. So we did that for all of our target weed species except for fleabane. One of the reasons we did that was to get uniform growth stages across the board. Once we'd germinated those weed species, so the picture down in the bottom left corner here is showing sow thistle germinating on agar. They were then transplanted into trays. So the next photo beside that shows the trays that we used. And our target for each population was 50 seedlings. We didn't always get that due to poor germination or lack of viability in the seeds, but that was the target. Once we transplanted those seedlings, we then allowed them to grow to their target growth stage. So for the broadleaf weeds, for example, it was kind of that 2, 3, 4 leaf stage. And for the grasses, it was, again, 2 to 3 leaf stage. Once we reached that growth stage, we then applied the herbicide in our spray cabinet at the recommended lower rate, I believe it was, of herbicide. And then this last photo here, we then made an assessment of whether individuals from that population were surviving and hence resistant. Also in this last picture here, again, it's of sow thistle. In all of our test runs, we included, where we had available, a resistant and susceptible standard. So that made, it just ensured that our herbicide was being effective and was killing the susceptible control, and that the resistant control was surviving. As we were doing the physical survey, so the collection out in the field, we were also taking note of what other weed species were present. And I've just put down here a little bit of a table looking at the species occurrence. Now, this is only in the Queensland surveys. So in the first column, we've got the weed species. The second column, we've got the number of fields where that target weed species was present. And then in the last column, I've put it as a percentage of the fields that we surveyed. So you can see that our most common weed species in terms of the number of fields infested was common sow thistle, which is no surprise. It's a weed that occurs in both winter and summer. However, in terms of the percentage of fields surveyed, wild oats was very common. I'm not going to go through this whole list, but I did want to point out one of the other weeds that we encountered quite commonly, and this was only in central Queensland, was sweet summer grass. So in most of the paddocks we surveyed in central Queensland in summer and winter, we got sweet summer grass present. So you can see there was a 34 fields had that species present. It was the only one really that wasn't on our target list that we noted was present in larger numbers. And I think John hopefully will mention later, we have a new project with investment through GRDC where we'll be doing another survey, and we've included sweet summer grass as a target in the northern region. Okay, so I wanted to move on now and talk about the results from the survey. I'll point out here that these are the results for the whole northern region. So these are including samples from New South Wales. The key resistance that we found in the northern region was to glyphosate. You can see there, out of the fleabane populations that we tested, every single one of them was resistant to glyphosate. Now you might say, well, glyphosate's never really worked on fleabane. We do have populations that we include in the testing that are highly susceptible to glyphosate. So that was something that we found. Also in relation to feathertop rosegrass, similar story. You might say, well, glyphosate doesn't work. We got 68% of our populations were resistant or not controlled with glyphosate. A similar percentage of our windmill grass. Noting there, we didn't have a lot of populations of windmill grass in our screening. For barnyard grass, 36% resistant. For sow thistle, we had a large number of populations tested. We got 14% resistant. Now, there had been a previous survey done on glyphosate resistance in sow thistle. It's going back 6-7 years now. And that was done by Annie Rutledge, and she did find a slightly higher percentage of resistance. I think it was in the low 20%. So this kind of shows that we haven't seen a huge increase in glyphosate resistance in sow thistle. Glyphosate grass, we didn't really find it as being a key species. We only got, I think we got a few more than 3 populations, but only 3 that had viable seed. And out of that, none of them were confirmed as glyphosate resistant. However, I believe, again, there has been a previous confirmation of glyphosate resistance in liverseed grass. In terms of other resistances that we found, out of our sow thistle, we had 95% were resistant to chlorsulfuron, and that's not really any surprise. But what is, I guess, alarming and maybe the start of things to come is that we did get one feathertop rosegrass population that was resistant to Haloxifop. And that's something I think In some ways, it wasn't a surprise, but it is a surprise. I say it wasn't a surprise because we've been expecting there to be Group A resistance in our summer grasses. I'm kind of surprised that we only got one population. This population was from up in central Queensland. And again, this is probably not a surprise. Central Queensland is kind of the home of feathertop rhodesgrass. Up in CQ, it's also a weed species that can occur pretty much all year round. So they've probably had a higher selection pressure for resistance towards this herbicide. Just moving on then to which herbicides are still working. So for sow thistle, favorably, we found that 2,4-D, we didn't have any resistance, so that's a very good thing. Also, we found that for fleabane as well. So all our populations were susceptible there. For the group A's, if you look at feathertop rhodesgrass, most populations were still resistant to Haloxifop and all were susceptible, all populations still susceptible to Haloxifop, and same with clethodim and paraquat. And then for ornless barnyard grass, we also didn't find at this stage any group A resistance. So that is a really favourable result out of this survey. A lot of group A selectives are going out to control these summer grasses. Hopefully they're going out in accordance with the recommended use pattern, so being followed up by a double knock to control any survivors. So at this point in time, it's a really favourable result that we've got most populations susceptible to our group A's. However, I think something to look out for. Also there, we did screen feathertop rhodesgrass for paraquat resistance and found none of that either. So that's very favourable, for that weed. I just want to now quickly show you just a couple of maps showing where we've got the glyphosate resistance in both common sow thistle and the next slide is on barnyard grass. I'm showing this because for both of these species, glyphosate resistance, I guess, is a developing issue. So there's a lower proportion of the populations had this resistance. So for common sow thistle, you can see it wasn't really based on any specific region. It is spread throughout the whole northern grain region, possibly less of it up in central Queensland, but basically across all our farming systems, we've got glyphosate resistance in common sow thistle. Again, it's an all year round weed species. Glyphosate's being used very heavily in our fallows. So whether it's a winter fallow or a summer fallow, the likelihood is there's a selection pressure being imposed on our common sow thistle populations. Moving on then to look at ornless barnyard grass. Again, it's a little bit of a similar scenario. Irrespective of where the barnyard grass was collected, there are populations that have glyphosate resistance. However, there seems to be a little bit of a shift in that it's more on the Queensland side of the northern region where we're getting a higher proportion of populations with glyphosate resistance. And again, I guess having it present in summer and in our summer fallows is why we've got such this increase in glyphosate resistance in barnyard grass. So in summary, I just wanted to reiterate, I guess, the points I've already pulled out. Glyphosate resistance is by far our most common herbicide resistance issue. And I only see it as increasing. Glyphosate is still being used quite a lot for fallow weed control. Sometimes that will be used as part of a double knock. Other times it won't be. It's something though that we'll have to keep a good eye on. We did get our first case of Haloxifop resistance in feathertop rhodesgrass. Again, I see this as probably the start of things to come. So that will be again a focus of our follow-on surveillance project. No 2,4-D resistance in fleabane or sow thistle, that's a favourable finding, and no identified paraquat resistance. As I did mention earlier, we'll be ramping up our screening of paraquat against a range of our summer grass species predominantly, in our future project. All these results have been sent to landowners. So something I didn't mention at the start is we sought permission from landowners in order to collect samples. The benefit of that is we have contact details for all of them, so we're able to send the results to them so that they can make more informed weed management decisions. There is a follow-on project with investment from GRDC, and I'm hoping John might give a bit of an overview on that. It is a national project which will be led again by John Broster. So I've kept it pretty short and to the point, but now's an opportunity if anyone had any questions or any points of clarification at all.
Okay, Michael, so you've got a couple of questions in the chats section.
Okay, now I'm not seeing the chat, so...
Oh, yeah.
No, for some reason.
Stop sharing, you might be able to.
Okay, hang on, I'll stop sharing. Okay, here we go, there they are. Okay, so there's a comment here from Mark Congreve about cross-referencing the Group A resistance level with the cotton industry survey, which shows a significantly higher level of Group A resistance. Yeah, so thank you very much for that, Mark. I am aware of the survey that's been done with the cotton industry. So what we might do is check and see whereabouts those populations were collected from. And maybe even something we might be able to do is to include some of those as our controls, our resistant controls in our future testing. So that might just strengthen our results as well. The other one here is from Bagarath. What were the doses of herbicides, lower or higher recommended? Actually, I stand corrected. I believe they were the higher label end, Bagarath. Yeah, I've just been doing some other work where I've been using lower doses. It's been at the higher label recommended. I could find out and get back to you in terms of the specific dose. Is that all right?
I can answer that for you, Michael.
Yep, have you got that, Adam?
1,050 AI per hectare, gram AI per hectare. I think we use a 1.5 liters per hectare rate for 2,4-D. Yeah.
Yep. Okay. Thank you for that, Adam.
For 2,4-D, we use all 1.5 liters per hectare. For glyphosate, mainly we tested it at 729 grams active per hectare. I think that's equivalent to 1.35 liters per hectare of glyphosate 540.
Yep, this is all clear.
Okay.
Thank you.
Okay, thank you.
Yeah.
And then there's just another comment there from Vicky. So Vicky Green, I'm guessing, that South Thistle and 2,4-D were also in the cotton survey. Yeah, so what I might do, I'll get in touch with Eric and cross-reference those.
Just a quick question, Mark. When you say higher level of Group A resistance in cotton survey, is that from the most recent one results from Eric?
Yeah, that's what I was talking about. So I think it was 2019 collections, something around there, might have been 18. And I think he got, I'd have to pull up the data, which I've got on file somewhere, but I think there was about 6 or so populations they come across, and they were tested for Haloxifop, Clethodim, might have been Propaquizofop, and maybe Quizalofop. They did multiple different Group A's, and there was differences between the fops as well, which was interesting.
Yep. Yep.
Okay, there's another one here from Henwen. Considering the high incidence of glyphosate resistance in fleabane, is there any need of using glyphosate plus 2,4-D as the first knock?
Well,
I guess
it depends on the growers, I suppose, because a lot of them just mix them up to hit both grasses and the broadleaf.
Yeah, exactly right. You're not always just going to have your fleabane present. You're going to have mixed population or mixed species of weeds. I guess if you only had fleabane present, you would reconsider whether or not you needed that mixture.
Particularly when you add on to that you've got a level of antagonism between glyphosate and 2,4-D. So if you're in a susceptible population, you're not going to see it. But if you're starting to get a population that's got a level of resistance in there, you're more likely to identify the incompatibility between those two.
Just a quick one, Mark. Correct me if I'm wrong. I think glyphosate is not registered on fleabane on its own.
Yeah, I think you're correct there. I don't think there's a straight glyphosate registration on fleabane.
It's always in a tank mix, if I'm not mistaken.
Well, yeah. You know, there's data sets out there that, you know, you can show additive effect, but there's also seen data sets where you'll show antagonism between the two.
Yeah,
so chemically there's antagonism, but, you know, mostly with that low level antagonism, you can overcome it by application rate.
Yeah.
Yeah, look, it's Laurie here. Can you hear me?
Yep.
We did some work years ago where we tried either mixing that glyphosate or 2,4-D or putting one before the other. So sometimes the 2,4-D first or the glyphosate first, up to, you know, I think we went out to, you know, about 4 days apart, and we consistently got a better result by mixing the two together than splitting them, like a lot of growers do to avoid that antagonism. We were targeting fleabane, and like I say, we certainly got a consistent improvement when the two were mixed. So I think that's where that has come from.
Laurie, on that, was there any like issues with the mixture if you mix, you know, generic round glyphosate with a different 2,4-D from product from other companies, or like some companies they do promote their own glyphosate and Roundup mixtures? Do you find any issues between different products from different companies when you mix them together?
No, look, we avoided trying to compare products from the different companies. We have done some work, but not looking at that mixture from memory. That would have been straight 2,4-D formulations. I think it was against fleabane, but not the mixture of glyphosate 2,4-D. Yeah, once you start getting into looking at the different companies' products, you really get a confusing message.
Yeah.
So yeah, we've avoided doing that. Yeah.
There is one more question here from Gulshan, and just asking about the reason for Haloxifop resistance in FDR. So Gulshan, if you're talking about the molecular side of things, I have no idea, and it won't have been looked at just yet. But if you're looking at why this has developed, and especially up in CQ, again, I think because glyphosate's not reliably consistent in terms of its control of feathertop Rhodes grass, Group A selectives have been used not only in their intended use pattern, for example, in broadleaf crops, they've also been pulled out and being used in fallow. And I think up in CQ, that has probably happened a lot sooner than it has when you move further south into the northern region. And I guess, again, it's just that selection pressure and exposure to Haloxifop. Look, I can't say whether or not it's been applied as part of a double knock, but that's why that will have developed first up in CQ. And Bagrath, looks like you're doing some work on Fly and 2,4-D antagonism on feathertop. And
yeah, this is just for implements. I think it's not recommended, but on grass, but initial our results showed that there is some kind of antagonism, but I need to confirm. So there, where we used glyphosate plus 2,4-D, the growth is much better, maybe 50% higher than only glyphosate only. Okay, so I'll confirm, but I think this is what we are doing at the moment.
Okay, excellent. Thanks for that.
I didn't know about that there is antagonism on grass also. So it's mainly because we use on broadleaf, but here we are testing on feather-top Rhodes grass.
Okay, thank you. Any last minute questions? No, Michael, I think that's it.
All right, Michael, thank you very much. And yeah, we're all giving you a round of applause here, but everyone's on mute, so you can't hear it. Yeah, no, that was good. Very well done. And it's, as I said before, that's our first comprehensive survey of the summer weed species of the northern region. So obviously some excellent data there, and you've generated some very good feedback there too. So that's excellent. Just a reminder that we are recording these sessions, so we'll be able to capture those chats as well as the presentation, so that we have it available for others who are unable to join. So yeah, just before I introduce John, just a reminder that there is a new herbicide resistance survey project that has just started this year. And this time it is a national survey, so the entire wheat belt of Australia is about to get surveyed. So people will be crawling all over paddocks around the countryside looking for weed populations. So these guys involved in the survey, John's team will be looking for farmers willing to allow the surveyors to come onto their property. So if anybody's got some growers out there who you think might be interested in getting involved, then by all means forward the details on to John and Michael and others. All right, so now introducing John. So John, are you ready to take over with your presentation?
Yep, I'm ready. I found one.
All right, so, start screen share whenever you're ready, mate.
Is that showing the right screen?
Yep, very good.
Right. There was a few of them going. Yep, so this is just looking at the results of the latest surveys across the various regions of New South Wales for the number of weeds we collect. Because I thought this was Thursday, not today, I haven't put the Queensland data in it as yet. But I've got that written down, so I'll be able to give that. So, right, so basically, I don't know what Michael said, but the surveys are repeated every 5 years. It's to provide a more accurate results for the herbicide incidence of herbicide resistance rather than what's sent into testing services or targeted projects, because these also survey areas where resistance is much lower. It enables us to show differences between regions and states, because even though they haven't been nationally coordinated before, they've all regions have been going on this 5-year cycle. And for those areas, which is southern New South Wales, through Victoria, South Australia and Western Australia, where they've been repeated every 5 years, we can see the changes in the extent of herbicide resistance between those surveys. And I don't think there's any of that in this presentation. It's just the latest results. So there's where we've been in New South Wales over this 5-year period. That was the 2013 to 2017 ones. A bit odd, there's so the slopes, which is the red and the pink, have been redone since then. So we averaging, we aim for 150 sites for each of the southern ones, except for the slope, for the high rainfall, which is 75, and we got about 250 from the northern New South Wales survey last time. So what did we find overall? if we extrapolate back from the data from the other surveys, over that period, about 64% of the ryegrass that we've collected across Australia is resistant to the FOPs and the SUs. If we look at New South Wales, it's only slightly lower than that. We're 58% and 50%. We have much lower Group D resistance than South Australia and Victoria, where that sort of sets the scene for the Australian resistance. Our Group M resistance is higher than the average, and our DIM resistance is lower because of the Western Australian effect there. So overall, we've sampled 600 ryegrass populations from New South Wales, and that's the results we've found in the most recent ones. Where that looks in relation to the various regions, you can see while we were 58% over New South Wales, that ranged from 23% for the FOPs, the west. So basically you can draw a line from about the Newell Highway heading west towards Mildura, keeping south of the Sturt Highway, so the highway from Wagga through to Mildura to an extent. So it's only 23% there. That shows that it's less intensive cropping, rotations are probably wider, and just less herbicide use. If we look at northern, it's 44%, but we'll see where that fits in later. And the other regions, it's all around that 70 to 90%. The plains covers out into that lower rainfall area a bit. And for this last year, the 2019 survey where we did the slopes, or we did the eastern region, that was 96% in this survey for 2019. And every population of ryegrass we collected had surviving plants to the FOPs. We look at the DIMs, it varies between the regions. Again, the more intense cropping regions, higher input, higher yielding, the southern and the eastern have the highest level of DIMs. SU resistance, or the Group B resistance, is relatively even between the SUs and IMIs, about the same level, and to an extent follows what we find with the FOPs, except for the plains region where the Group B resistance is much lower. We had the highest Group D resistance in the slopes. That may be because the Group Bs have fired a bit earlier, and limited other resistance, no J and K or K, but M high in the north, showing the importance of glyphosate in that winter summer cropping region for the fallows. High in the south, probably because of the higher inputs and the wanting to get seed banks down for coming crop because of the lack of in-crop choices. But in other regions, less amounts. The east had a couple of populations this last survey. The wild oats, New South Wales, 34% were resistant to FOPs, as they were in Queensland. Western Australia, it's 48%. I thought New South Wales might have been higher than Western Australia because 90% of the samples we get for the testing service here are from New South Wales. We had only it looks like 2 populations resistant to the DIMs in New South Wales, and there's a few populations resistant to the SUs, but no resistance to J or M yet so we've still got some options there. This year's higher rain, or the last higher rainfall survey, had some Group B resistance in the wild oats, but we have limited when we go out into the lower rainfall regions. If we look at the various regions, as I said, much lower in the west, again, that lower intensive, but we noticed where we had lower levels of resistance in the ryegrass, in the FOPs, so the north and the plains, we have higher levels of resistance in the wild oats, most likely because it becomes more of a major weed and it's the targeted weed in that region. The Group B resistance, you'll notice in 2014, it was 53% in the high rainfall regions. We're getting better at working out what's alive and dead now. We probably were a bit pessimistic then. It's about 20%, but it's still the highest region, showing the higher use of that. Herbicide groups for wild oat control. Brome grass, some resistance, not much, a little bit to the group A's, the FOPs, and a little bit to the SUs and the IMIs. Highest in the west, where in a lot of those, when we went further west, it was one of the dominant weeds. We found the further west we went, we were more likely to find the brome grass and the barley grass than we were ryegrass. So we can show that here, it represented with the Western region surveys. Barley grass, yes, we've got the only FOP resistant ones from the west, the plains, so going out to the northwest of the southern region, and the west had significant Group B resistance. What we've got here is we're finding the occasional population of barley grass resistant to paraquat. I think of the three or four populations, because not all have been tested to it, the majority of them have come from loosened pastures. So that winter cleaning option, and we seem to be getting a bit of paraquat resistance. We've got a couple of paraquat resistant ryegrass populations, not from the surveys, but from the testing service themselves. We look at wild radish. So we're going on to the broadleafs now. We don't find as much of that in New South Wales. It's probably our number 2, maybe 3 broad leaf weed. We look at it compared to Western Australia, where it's their number 2 weed. Our levels are much lower, like 14% resistant to the Group Bs, 12% to Group Fs, so diflufenicin, compared to the numbers of 80%, 50% in the West. So we're just showing the less importance of that weed species. South thistle is our major of the broadleafs that we find, as you can see from the number. About half or more than that of the south thistle is resistant to the SUs. We have the occasional one resistant to the phenoxys, and all of the glyphosate resistant ones have come from the northern region and the northern survey. And there's a few from Queensland as well that I haven't got in this report. And the other weed species we collect is Indian hedge mustard, just a few more than the wild radish. And we're getting a bit of Group B resistance in that. And at a couple of surveys, we've probably got one population resistant in the north to 1 or 2 resistant to diflufenicin. Probably not all of them were tested. And 1 resistant to the phenoxys further south. So that's the results of the survey. But thank GRDC and MLA for funding them, and the numerous casual staff and students who I've utilised as travellers and cleaners and pot fillers over the time. So probably any questions now?
So John, as you probably picked up on from after Michael's presentation, we're going on a combination of chats.
Yep.
So if you have a look in the chat section, you'll see some questions there. You've already got one from Han Wen.
Yep. Yeah, so with that, we've got 3 populations of ryegrass now. One from, maybe all 3 from southeast South Australia, where the small seed growing takes place.
did you want to just talk through the results for Queensland that you didn't have a chance to include in your slides?
Yes. So, well, the only one I did the wild oats, which I've reported, and if I for some broadleafs, which we did. So we found, and this is a mix of wild radish and what have we got? A mix of wild radish, Indian hedge mustard, turnip weed, African turnip weed, and a few we didn't know anything about. Like we weren't sure, so we filed them under miscellaneous. What we've got is about 11% of them are resistant to the group Bs. So that was 64 we had. We had no resistance to Roundup. We had 19% resistant, or about 8%, sorry, resistant to the phenoxies. We've got about 20% of 16 that we tested to the 9 of 30 that we tested to diflufenicin were resistant, no resistance to atrazine, and no resistance to Innovix, so the IMIs. So we've got a bit of SU resistance, diflufenicin, and phenoxy resistance amongst those.
Okay, I found this on the web for resistance and
Shut up, Siri.
So that was just in a general group of brassicas, was it, John, from Queensland?
Yeah, from Queensland and northern New South Wales. So mainly, majority of which were turnip weed, limited seed availability. So it looks like the group B resistance is spread across every species except the African turnip weed.
Okay, so you got a couple more questions coming through on the
Yep.
chat folder.
Always interested in ryegrass populations, Bagarath. Yeah, it wouldn't surprise me that in that irrigation area, what I've found before when I looked at a Shire basis for glyphosate resistance, that the higher the proportion of summer cropping or irrigated cropping, the higher the proportion, the percentage of glyphosate resistant populations you get in ryegrass, because you've got summer cropping in the irrigation and the summer cropping regions with winter fallows where you're treating your ryegrass with the glyphosate in the fallow period where they've got no competition to help. Yeah, and Michael Widderick, it was 34% for the FOPs for the Queensland wild oats. I had that up on the slide, I did. And 3% for the SUs, no glyphosate and no Abidex trilate.
All right, that's good, John. Very good. Yeah, I was just going to say that as well, Michael. So yeah, you got a question from Gulshan there, and then probably wrap it up with maybe another reiteration of the national project and need for sampling sites.
All right, so resistance factor for Avidex, I'm assuming you mean how much is there? We haven't found any in Australia. Where they've found it in Canada, it's sort of been 10, 15 years of continuous use, like every year that it's been necessary to develop resistance to Avidex. There is resistance in the ryegrass to Avidex in South Australia and Southern Victoria now, where they've been using it either as a combination with trifluralin or as an alternative. So it will probably arrive in the wild oats, but it's one of the rarer ones at the moment. Yeah, so for the national survey, yeah, we've got it. We're aiming for each of the, in New South Wales and in Queensland, the GRDC sub-regions. The aim is to get 150 paddocks sampled in each of those. That allows us to detect a resistance at, if it's present at 1%, I think, of the populations, on a 95% likelihood of getting it right. So yeah, we're looking for a spread of paddocks across those regions, so we can get wide range, so we can then do follow-up, and then for the next round of surveys, we're going back to the same paddocks, or at least the same farms if those paddocks aren't in crop. So yeah, if anybody's got any people they know of they'd like to be involved of in New South Wales or Queensland, let myself or either of the Michaels know. So that's about it, I think.
No, that's very good, John. Yeah, and I will just, I guess, add that this survey is a major undertaking. I think you're looking for something close to 1,000 paddocks across the northern region. Yeah,
so yeah, it's I think it's 1,050 across. If there's 6 regions, it might be 1,200. 6 regions in New South Wales, sub-regions, and 2 in Queensland, I think we're looking at. So yeah, a lot. Yeah, it's substantially more than has ever been done before, in one hit at least anyway. So it is a big undertaking, so need a lot of support. And you've just got a question there from Laurie. There is a summer weed portion of it. I think is it 3 regions, Northern New South Wales and Queensland, looking at, I can't remember offhand, Michael Widderick might know of how many, if it might be the 150.
Yeah, I think we've got 250 fields to be visited during summer 2021.
Right.
Yeah. And I think our target species, I've got the file up in front of me here. Let me just find it. So in the northern region, our species to target are annual ryegrass, wild oats, feathertop, hornless barnyard grass, wild radish, fleabane, sweet summer grass, and south thistle. One of the points of difference between the previous survey and this new survey, there's going to be a bit more coordination nationally. One example of that is, for example, all fleabane populations collected nationally will be tested in Toowoomba. So where in the past, John, Michael and the group at DAF have worked together and shared samples just between the northern region, there's now going to be sharing of samples nationally as well. So that's a bit of a point of difference. One of the reasons we're doing that is to have a bit more uniformity in terms of our results, because they'll be tested all at the one site for any particular species.
Yeah, so it's a 3-year project. So we've got 1 year to collect everything and then 2 years to screen it. Results will go back to the farmer themselves, possibly when they become available, or definitely a full report at the end of the project for all weeds collected in that farmer's paddocks.
All right, John, I think on that note, we'll wrap it up. And yeah, thank you again to John and also to Michael. And thank you all for joining the first of the 5 seminar series. And hopefully you can join us again next week. Have a good day, everyone. We'll see you next time.
Thank you.